Okay, so last week we focused almost exclusively on very straightforward monohybrid crosses where we were looking at heterozygous versus homozygous, dominant versus recessive, and being able to figure out the phenotypes from the genotypes. So this week we're going to move into more complicated material, and we're going to start with some of the trick crosses that you might see on exams. So the first one that we're going to talk about is the Punnett squares where progeny die. So you'll have some standard setup, right? So like in this case, let's say I have a disease B is caused by gene B, and you only have the disease when you have the recessive, you're homozygous for the recessive allele. But people who are homozygous for that recessive allele do not survive development. And so the question might say something like, what is the ratio of heterozygotes in the living progeny in a cross between two heterozygotes? So what normally happens when people run across these problems on exams is they'll read straight through it and they'll kind of burn past that the question is asking only for a percentage within the living progeny, and they'll give their usual ratios. So let's go ahead and set up this cross. So okay, it's a cross between two heterozygotes, and if I'm heterozygote, that means my alleles are not matching, that I've got one set of alleles that's uppercase and another one that is lowercase, and so both the parents are heterozygous, so I will set that cross up like that. And so again, to do the monohybrid crosses, you're just bringing the alleles in, so this progeny would be big B, this one would be big B, little b, the heterozygote. This one would also be a heterozygote, and this one would be homozygous recessive. So under normal circumstances, if they were asking for the ratio or the percentage of heterozygotes, you'd be like, okay, well, there's 1, 2 out of 4, so that's 50% that are heterozygous. But the question is specifically asking only for the living progeny. And so if you see something like that, you find the ones that do not survive development. So in this case, it's people who are homozygous for the recessive allele, so that would be this one here, and go ahead and cross it out, because that will help remind you that your ratios have now changed. So after you cross them out, now I'm looking for the ratio of heterozygous in the living progeny. So these did not survive development, so only these three made it. So my two heterozygotes are now 2 out of 3. They're not 2 out of 4 because the question only wanted the ratio in the living progeny. So it would either be 2 out of 3 or 66% would be how you handle those crosses. So when you see them on an exam, be sure that you're reading the problem really thoroughly and you're very clear on what exactly they are asking in that question. So don't give them the ratios that they're not looking for. The other thing that you might see is something that I call a reverse Punnett square. So in these examples, you're given details about one of the children, one or more of the children, and you're asked to figure out the genotype of the parent. So in this example, we're going to say that we have a gene Y in plants that controls color, that the yellow color is dominant to green, and all it's going to tell you is that all progeny are heterozygous for this Y allele. And so, okay, if they are all heterozygous, how I start these is I start filling in the chart. So I always draw my Punnett square, even for the reverses. So all progeny are heterozygous, so that means in each one they're going to be, again, the mismatched alleles, one dominant and one recessive, and I'll do that for all 4. And then to solve these, you can literally just take one allele back on each side, right? So let's just assume that this big Y came from this parent. If we just drag it back on this row, and we can also guess that this Y went back for this parent. So one of the parents has to be a homozygous dominant, and when we repeat this for our columns, so in both this case we're going to say that the smaller Y came from the parent up top in both cases. So now I know my genotypes for the parent is homozygous dominant for one and homozygous recessive for the other. So most of them will not be that clean cut, but let's look at another example. So in this one, freckles are controlled by gene F and freckles are recessive. If one sibling has freckles and their sister does not, what are the possible genotypes of the parents? So again, still draw your Punnett square because it will help you. And then sometimes you can guess at the genotype of the children. So I have this child who has freckles, and they're saying that freckles are recessive. So I must have 2 copies of the lowercase allele in order to have a child with freckles. So this person, I'm going to just go ahead and put them in the lower right corner. That's how I like to do things. I'm not sure it exactly matters. And at least one of their siblings does not have freckles. So you may be tempted to put big F in here, but remember that not having freckles is dominant in this case, and so you can't really tell what their other allele is going to be. They might be homozygous dominant or they might be heterozygous. And if you don't know, you can't really say. So we can make some guesses about the possible genotypes of the parents. So if I have a homozygous recessive child and I just drag, one allele back from my row, well, parent 1 has to have at least 1 recessive allele. And I can do the same thing for my column. Parent 2 up here also has to have at least 1 lowercase allele. You can't get a homozygous recessive any other way unless the parents are at least each carrying that lowercase allele. Now on this side though, this uppercase F, that dominant allele, had to come from somewhere, so we're going to just randomly assign it to this parent here. So one of the parents has to be heterozygous. It cannot be any other way. It might have come from up here or this side, we don't know. And because of that, for the other parent, they could either be heterozygous or homozygous recessive. So those are possible genotypes of the parents. So now we're going to move to genetic diseases. So there are around 25,000 genes in the human genome, and previously we've been talking about traits like hair color or skin color, and when there are mutations in those genes, yeah, you look a little different, but it's not that much of an impact on you. There are many proteins that we need to survive, that we need to stay healthy, and when we get mutations in the genes that code for those proteins, you end up with these genetic diseases. So like I told you earlier, most mutations have the effect of breaking a protein. And so as long as you have 1 good copy, you're probably good to go. So most of the diseases that we're going to talk about are actually going to be recessive. And most of our chromosomes 1 through 22, right? There's just more genetic information that is stored on those autosomal chromosomes, not that much on the X and Y chromosome. So most diseases are going to be autosomal recessive, but there's one example of a dominant autosomal disease. So the disease that we'll talk most in depth about is sickle cell anemia. So sickle cell anemia is caused by mutations in the HBB, hemoglobin beta gene. And if you remember back to the beginning of the semester, we talked about how hemoglobin was this big protein whose whole job was to deliver oxygen to the various parts of the body. So it is obviously really important for our functioning and for our life. So the HBB gene is found on chromosome 11. And because it's on 11 and not the X or the Y, it is autosomal. And mutations in this gene are autosomal recessive, meaning you need 2 copies of the bad gene to actually have the disease. So S's are sort of hard to tell apart, and so are C's. So I'm going to recommend that we stick to using the A alleles. And so just to go through the genotypes that you might see in sickle cell anemia. So the first genotype is going to be homozygous dominant. So this is a healthy non-carrier. So most of us right now, we probably have 2 good copies of the HBB gene. We're probably healthy non-carriers. If you're heterozygote for a genetic disease, we have a special word for that. We don't call them heterozygotes anymore, we call them carriers because they are carrying around a bad allele for a gene. And in this case, this is going to be a carrier of sickle cell anemia because they are heterozygote. They are carrying around that disease allele even though they themselves would appear healthy. So if you look on an exam and it's saying someone is a carrier for sickle cell anemia, you automatically know that this is their genotype. They have the heterozygous genotype. And similarly, somebody who actually has sickle cell anemia is going to be homozygous recessive because they have 2 bad copies of that hemoglobin gene. So what is actually happening in sickle cell anemia is that the mutation causes a change from a glutamate amino acid to a valine amino acid, and that makes the protein really sticky in low oxygen conditions, which causes the red blood cell to actually be sickle-shaped. And when it's sickle-shaped, it cannot do its job of delivering oxygen, and it will also get stuck in like your capillaries, or your arteries, where it's going to cause stroke, all sorts of problems, right? So the symptoms of sickle cell anemia are things like joint pain or shortness of breath when those red blood cells aren't able to carry oxygen. People are often fatigued because they're not able to get oxygen into their tissue. There's a lot of pain, they have a lot of infections, but the worst part is that they are probably, their lifespan is expected to be in the mid-40s. So what actually happened, and this is one of the mutations that we do have to know in depth, is that if we have this double-stranded DNA section, so this is the real start of the HBB gene and its normal form, and so we have our sense strand 5' to 3' on the bottom and the antisense strand 3' to 5' on the top. So that gets converted into mRNA, and that mRNA is then translated into protein. So that's the ribosomes coming in with those tRNAs. And everything that we're going to focus on is actually happening in codon number 6. So in codon number 6, I have a sequence of GAG, CTC, that's changing to GAG in our mRNA, and that GAG will code for a glutamate. In the sickle cell form, so we're only going to look at one allele. This is a big gene. We can have mutations anywhere, but we're going to focus on the disease allele that has a mutation in codon number 6. So instead of being GAG, now it's GTG. And so any change in the DNA will cause a change in the mRNA. So instead of GAG in the mRNA, we're going to have GUG. And instead of a glutamate, now we're going to have a valine. So remember that the proteins are just a chain of amino acids. So this is the whole structure of hemoglobin, and then this is the section that's actually coded for by the HBB gene, and it's this part right here where the mutation is occurring that's causing sickle cell anemia. So what is happening is that we're swapping a hydrophilic glutamate. So this is an amino acid. We've got our amine group here connected to a central carbon and a carboxyl group on this side. And then we've got this R group. And this is what makes it glutamine. So this is hydrophilic because of those oxygens. They want to make friends and hydrogen bonds with that water. So this one turns into a valine. And this valine has nothing but carbons and hydrogens. So just like the lipids, it's hydrophobic. And under low oxygen conditions, you go from a normal red blood cell to one that is sickle-shaped. So you do have to know these details, that it is a single base change, GAG to GTG in codon number 6, which is causing a change in the amino acid, in that 6th amino acid, from glutamic acid to valine. So this causes that hemoglobin to be sticky under low oxygen conditions and will eventually give it that sickle cell shape, which causes all the other problems that you see in sickle cell anemia. So that's one genetic disease. The other one we're going to talk about is cystic fibrosis. So cystic fibrosis occurs because of mutations in the CFTR gene. So CFTR is a chloride transporter that we find on chromosome number 7. So again, because it's not an X or a Y chromosome, this means that it is autosomal, and like mutations, this one is going to be recessive. So the alleles that we're going to use for cystic fibrosis are going to be F. I'm trying to find a good letter for this one because C definitely doesn't work. It's too easy to get confused. So when you're writing these, make sure that your big F and your little f are very distinct because it is easy to get confused. So again, we're going to use that same nomenclature. A homozygous dominant means that you are a healthy non-carrier, and we're not calling heterozygotes anymore for diseases. So the heterozygote for the cystic fibrosis allele, we're going to say they're a cystic fibrosis carrier. So if you see that description in a problem, you know their genotype up front. And a person who has 2 bad copies of that CFTR gene is going to be somebody with cystic fibrosis. So The CFTR gene, like I said, it encodes a chloride transporter. So this is a membrane protein whose whole job is to move chloride outside of the cell. When there are mutations in that gene, it can't work, it does not function. And so chloride tends to stay inside the cell, and this interferes with osmosis. So what ends up happening is, instead of having a watery environment outside the cell, mucus will build up on those tissues. So this has the effect of causing increased infections. People with cystic fibrosis definitely have trouble breathing, and if you see pictures of them online, they also look really skinny. And that is partially because mucus is also forming in their pancreatic ducts, which will impair that function, so they are not able to digest food well. And people with cystic fibrosis do unfortunately pass away, usually by their early 20s. So this is really not an awesome disease to have. So these diseases work like normal inheritance. So in this case, we've got two heterozygotes, so two carriers. So mom and dad are carriers, and then you would just set up the cross the same way. So in this case, we have 50% that are carriers of the children, 25% that have sickle cell anemia, and then 25% that are healthy non-carriers. So that's cystic fibrosis. So Huntington's disease is the only disease that we'll talk about that is a dominant genetic disease. So the nature of the mutation is a little different. The ones we've been talking about have been single base changes. So this mutation is going to be a little different, which is why it ends up being dominant. So Huntington's disease is caused by mutations in the HTT Huntington gene. So we don't really know what the function of Huntington is exactly, but we do know that it is expressed highly in neurons. And this gene we find on chromosome 4, and again, it's not an X or a Y chromosome, so this one is going to be autosomal, but in this case, the disease allele is dominant. So we'll use letters big H and little h to represent our alleles. Someone who is heterozygous has the disease. I don't think we see many homozygous dominants. I'm not sure that they survive development. And someone who has two lowercase h's has or does not have disease. They are healthy. So this is flipped from all the other ones that we've been talking about, right? So in Huntington's disease, it's not a single base change. What's actually happening is that there is a CAG triplet repeat kind of near the start of the Huntington disease, and this triplet repeat does tend to be replicated like it's expanded during DNA replication. So it kind of causes the DNA polymerase to stutter and add more CAGs onto it than it should. And so this causes the protein to be really sticky and run around and get entangled with all the other proteins in the cell. So when that happens, the neuron can no longer do its job, and those neurons will begin to die. So people with Huntington's disease, it usually shows up when people are in their 40s or 50s. They have things like dementia or delirium. They basically need hospice care. They can't walk, they can't feed themselves. It's a really bad disease to have. And what's worse is that, again, if you don't find out that you have it until your 40s or 50s, chances are very good that you already passed it to your children. So this is just a look at some neurons. So in this case, this is wild type, and the little neurons, they look pretty happy, but when they have the Huntington gene expressed, they get full of protein and they're going to start to die off. So it does cause brain shrinkage, which is not a good one, right? So when you're setting up the crosses, so say you have a father who has the disease, in which case he would be heterozygous, and you cross it with a mom who does not, half the children usually, right? Because this is a dominant disease. You only need one allele to actually have the disease. So in this case, half the children will have the disease.